Miss How? Yao? Was it How or Yao?
Yes, Doc.
Yes. How many are in already? Yes. Yao. Yeah, that's right. How many are in already?
111 po.
Okay. So anyway, you have still 1 minute before time. So you take note after 15 minutes. Okay. Okay.
Yes,
And
Doc.
yes, class, kindly change your profile. If it is about the comics or what you call this, not your picture. If it's cartoon or whatever, please change it. I want to see your actual faces. Okay, class?
Po, Doc.
Okay,
Doc.
what are these that I see here? Okay, and as much as possible, the photo should be the one you submitted in the during the interview. Oh, it should be a formal photo, okay, that I can see your, I can see even if you have pimples. Okay, no, for example, okay, that's it. You should be in uniform. I'm sorry, yeah, 1 month already, no? You should be in uniform next time because it's already 1 month. Okay, so please, no? We started supposedly what time? September 21.
21, Doc.
Okay, please. Next time. Okay. So I'll just check if there are still. Oh, this one. Oh, who's this? Okay. Plisuela, you don't have Plisuela era. You don't have any picture yet.
Hey, Doc, I just turned on my camera na lang po.
So you don't have yet picture.
I'm not sure, Doc, why is it why
Oh, Janelme Margalio. Huh? You are in a duster. I don't want that. Huh? You are in duster. So you should be in your uniform. If you don't have that uniform, in a proper dress with what you call this? Sleeves. Santos Noel, you have, okay, there you are.
Good afternoon, Doc.
Okay, yeah. Oh, this one is good. What uniform is that? Nursing or what? Anyway. Okay. Oh, we don't have X, no? Meron ba tayong X, the family name? Wala. Oh, you see, Cabello, beautiful. Oh, Miss Chu, beautiful. Okay. You change your, your Margalio. I don't like that. Hmm? You hear me, Miss Margalio? I don't like that picture.
Yes, Doc. I'm looking
Okay.
for a formal one pa.
Yeah, I'm looking. You have so many dresses. Insignia, Sinia, none yet. Uy, not yet. Oh, look at Geraldine Calvero. Very proper, no? It's good. Miraflores naman, happy, huh? That's good. Be happy now because you might be not happy at the end of the year. No, I'm just joking. Yeah, Gwendolyn Miraflores. Oh, who's this? Who's that comic dress? Huh? Okay. 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 Servano Maria, you don't have yet. This one, sino itong si Argil Kahilig? Huh? Who's Argil Kahilig? Huh? Okay, this good.
Na.
Next time I'll be checking your. I'll be
in my nursing uniform.
Yeah, oh, that's okay. Next time I'll be checking your hair. The gentleman.
Do you see the new picture, Doc?
Yeah, of course. I'm looking at each one. I have
Thank you,
time.
Doc.
Oh, eto, nung nag-graduate pa yata si Radam, meron pang QP, ano ito? Tacloban? Katrina. Oh, happy si Katrina. Okay. Okay. Alcover, Benedict, good. Dagami. Louis Sudario, you don't have a picture yet? Huh?
Not yet, Doc.
Why? You're still making a good picture, a poise?
Mimiling pa, Doc.
No, don't look for it. Any face is good so long as you pass the histology, diba? O kahit naman gwapo, kung hindi ka papasa, you're not gwapo at all, diba? Okay. Anyway, Aldaba, Leslie, you have no picture yet. Aldaba. Okay, next time. Okay, I'm telling you now. All of you must have pictures already. When I start, that should be completed. Otherwise, I'll give you a minus in your first examination. -2, -3, up to -5. Is that clear, class? Is that clear?
Yes, Doc.
Yes.
Yes,
Okay.
Doc. Good,
Doc.
Doc.
Yes, Doc.
Sige. Saak. Oh, Jessa. Where are you, Jessa? I haven't seen you for a long time. Jessa, answer me. Maasin? You're still in Maasin?
Yes po, adila ako nga di.
Sa inyo?
Yes,
Or di ka makauyab kung permi kang ada sa inyo.
Di ba, siring ni mo, Doc, dere, pwede pa.
First year dire pa. Okay. Anyway, we shall start. Okay. If we have time, I'll conduct a recitation, if no more than perhaps. But I think we will extend because Thursday we have laboratories. Since we do not have laboratories, we just send it by email. So I have the what they call this, the liberty to extend the our talk. Anyway, there is no lecturer for this afternoon. Okay, so let's start. Histology of blood. Okay. 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 Okay, medtech, I'm sure you know all this. Okay, our blood is about 5.5 liters in a man. Okay, consists of the cells and fluid that flow in a regular unidirectional movement within the closed circulatory system. That's why when you have injured yourself and there is blood oozing out, then there will, yes, there will be problems with your what you call your blood pressure and all. Blood is propelled mainly by the contractions of the heart. As the word says, mainly, but of course the other big arteries will also be used, especially when it is downstream. Okay, so you have unidirectional and mainly propelled mainly by the contraction of the heart, made up of two parts. You have formed elements or blood cells and the liquid, the plasma. Plasma is the liquid in which formed elements are suspended. The formed elements are the erythrocytes, the platelets, the leukocytes. Okay, now we will be going back to this always. So when you centrifuge your blood, you will have more or less two layers that you readily see, but actually they are three. Okay, so you have here and this here, the form elements, okay, red, and then you have your plasma. Okay, if blood is removed from the circulatory system, it will clot. This clot contains formed elements and a clear yellow liquid called serum, which separates from the coagulum. Blood that is collected and kept from coagulating by the addition of anticoagulants like heparin, citrate, okay, so they do not coagulate. When centrifuges, it separates into layers. Okay, now what is the value of this? The hematocrit, which is a laboratory procedure okay, is an estimate of the volume of packed erythrocytes per unit volume of fluid. The normal value is 40 to 50% in men and 35 to 45% in women. The plasma, okay, so this is your plasma, the translucent, yellowish, viscous supernatant obtained when whole blood is centrifuged. There you are. Now, the liquid in which the formed elements are suspended. So you see here the main two layers. You have your plasma and you have your red blood cell, okay? This is more dense, that's why it is below, no? It is down. And the buffy coat, which is composed mostly of the white blood cells and platelets. That is the buffy coat. So you have actually three layers: the plasma, the buffy coat, and the red blood cells. Composition of plasma. Let's go to plasma first. You have water, 91%. It is an aqueous solution containing substances of low or high molecular weight that make up 10% of its volume. The plasma proteins, 7% of the volume. The inorganic salts, 0.9%. Organic compounds, the remaining, okay? So the others would be the vitamins, hormones, and the lipoproteins. The low molecular weight components of plasma are in equilibrium with the interstitial fluid of the tissues. Take note of this, huh? The low molecular weight components of plasma are in equilibrium with the interstitial fluid of tissues. The composition of plasma is usually an indicator of the mean, okay, of the mean composition of the extracellular fluids in general. While this low molecular weight components of plasma is in equilibrium with the interstitial fluid of the tissues, while the other one is extracellular fluids, okay, in general. The plasma proteins. The plasma proteins are, first we have albumin, maintains the osmotic pressure of the blood. This is the most abundant plasma protein. And then you have alpha, beta, and gamma globulins. You have lipoproteins. You have proteins that participate in blood coagulation, the prothrombin and the fibrinogen. Okay, so as you know already, plasma is 55% of the blood volume. Okay, you have 91% or 90% water. 7 is the blood proteins, which is the albumin, okay, and the globulins and the prothrombin and fibrinogen, and the other remaining are the nutrients, okay, sugar, amino acid, lipids, hormones, and so forth. Now the cellular components, okay, you have your, of course, your blood, red blood cell, and with this buffy coat are your erythrocytes, okay, platelets. Functions of the blood: transports nutrients from their site of absorption or synthesis, okay, distributing them to various areas of the organism. Transports metabolic residues which are removed from the blood by the excretory organs. Serves as a distributing vehicle for the hormones. Permits the exchange of chemical messages between distant organs for normal cellular function. Participates in the regulation of body temperature in acid-base and osmotic balance. The blood, okay, transports oxygen, carbon dioxide, metabolites, and hormones, among other substances. Oxygen bound mainly to the hemoglobin of the erythrocytes. Now, carbon dioxide bound to the proteins of the erythrocyte, mainly hemoglobin, is carried in solution in the plasma as carbon dioxide or bicarbonate. Other functions of blood, okay? You can see here the features of basophil Eosinophil, neutrophil, monocyte, lymphocytes. We'll go at it one at a time. Leukocytes, one of the body's chief defenses against infection, circulate throughout the body via the blood vascular system. While suspended in the blood, they are round and inactive. Round and the word inactive. So when you have injury, that's the time that they become active, okay? So they do their functions. Its cell will do its function. Cross wall of venules and capillaries, these cells penetrate the tissues where they display their defensive capabilities. Blood separates into layers when centrifuged. The lower layer, as you saw already, represents 42 to 47% of the entire volume of blood in the hematocrit tube. It is red and is made up of erythrocytes. The layer immediately above, you call that the buffy coat, 1% of the blood volume. It is white or grayish and consists of leukocytes. These elements separate because the leukocytes are less dense than the erythrocytes. Covering the leukocytes is a fine layer of platelets, not distinguishable by our naked eye. Okay, that's part of your buffy coat. Okay, so. Okay, now let's go to the erythrocytes. Okay. They are anucleated, packed with oxygen-carrying protein hemoglobin, biconcave discs without nuclei, isotonic, medium. Human erythrocytes are 7.5 micron in diameter, 2.6 micron thick at the rim, and 0.8 micron thick in the center. The biconcave shape provides erythrocytes with a large surface to volume ratio, thus facilitating gas exchange. The normal concentration of erythrocytes in blood, you have 3.9 or 4 to 5.5 million per microliter in women and 4.1 to 6 million per microliter in men. Okay. Miss Aldava. Leslie. You can hear me, Miss Leslie Aldava? Say yes.
po, Doc.
Yes, very good.
Yes.
Okay.
Yes po, Doc. Thank you, Doc.
Okay. Yeah,
Yes
good.
po, Doc.
Thank you. There is only one Aldava. Okay. Okay. What is the purpose of this biconcave shape of the red blood cell? What do you think? No?
It provides more containment for oxygen po.
And it's here.
It provides more, and
Large.
it allows our BCs to, yes, large, and also it, will I read that lang, Doc?
Yeah, okay, you can have your own word so long as it has the same meaning.
Hello, Doc.
Yes. Okay. Here is the answer. Surface to volume ratio.
Yan po.
Okay. So meaning
large
yes,
containment for ano.
for containing gas exchange. Because like in your boyfriends, you know, with boyfriend, you have boyfriend, Miss Aldava. Are you the one? Where are you now? Yeah. You have a boyfriend? Uy.
No po.
Oh, baka naman
No po.
meron. Oh, so, okay. You will have more heat generated if more part of your body is what? Okay. You know, met with the other surface of your boyfriend's son. Diba? So that's it, the same. There will be exchange of blood. There is oxygen being given to the other side if there is a large surface of contact. Is that true, Miss Dava? Answer, yes? Okay, anyway. The normal concentration of erythrocytes in blood, as we mentioned
Yes
already.
po.
Okay, now another question to Miss Famador. Miss Famador, yes, Miss Famador, why do you have more, you know, concentration of blood in males compared to females? What do you think? Didn't you expect that you should have more because of your monthly visitor? What do you think is the reason why males have more, okay, greater concentration of erythrocytes in blood? Faster.
Probably, I'm not sure about this, Doc, but I'll try. Females have lower concentration of blood because of menstruation, and that's absent in males. So they have larger concentrations of
Yes. No, it's not
RBC.
related because that is physiological loss. Okay, that is not counted in your blood volume. What is here? The answer is the males are generally accepted to be more active, and therefore they would need more oxygen. Okay. Okay, that's simple as that. Okay, medyo active, but of course nowadays medyo hindi na siguro. Anyway, that's the main reason. Okay. RBC adapt to the irregular shapes and small diameters of capillaries. They can adapt because it's important that they could, provide nutrients. Okay, so there is a term for that later on. It's diapedesis. Anyway, erythrocytes containing normal adult hemoglobin are easily deformed and frequently assume a cup-like shape, as you see in the picture. Adult hemoglobin. Now let's go to reticulocytes. Erythrocytes recently released by the bone marrow into the bloodstream. So they are recently released. Often they contain residual ribosomal RNA, which in the presence of some dyes like brilliant crescent blue can be precipitated and stained. So you have that, no? The young erythrocytes with few granules or a net-like structure in their cytoplasm normally constitute about 1% of the total number of circulating erythrocytes. So if you have more than 1%, then that is already abnormal, okay? This is the rate at which erythrocytes are replaced daily by the bone marrow. So erythrocytes are basically replaced 1%. So because this is the rate at which erythrocytes are replaced daily by the bone marrow. Increased numbers of reticulocytes, take note of this, okay, indicate a demand for increased oxygen carrying capacity. Okay, perhaps you are in a high altitude area, so you need more oxygen, or perhaps your carrying capacity is less, so you should have more blood, red blood cell. But if the production is less or is not enough needed, so the reticulocytes are sent out. At least they can carry also oxygen, but in a minimal way. That's your reticulocytes. So you call that, but you have more than the normal reticulocytosis. Okay, erythrocytes lose their mitochondria, ribosomes, and many cytoplasmic enzymes during their maturation. Okay, that's why you have no nucleus in your red blood cell. The source of energy for erythrocytes is glucose, which is anaerobically degraded to lactate. Do not have a nucleus. Of course, the red blood cells doesn't have a nucleus or other organelles necessary for protein synthesis. They do not synthesize hemoglobin. Okay, the lifespan of your human erythrocytes is about 120 days. Worn out erythrocytes are removed from the circulation mainly by macrophages of the spleen and bone marrow. The signal for removal is the appearance of defective complex oligosaccharides attached to integral membrane proteins of the plasma lemma. If there are already your oligosaccharides, defective complex attached, then that shows that the red blood cell is old and there's a signal now to change them or, disintegrate and produce another red blood cell. Always, there is always production. In disease states, nuclear fragments containing DNA remain in the erythrocyte after extrusion of its nucleus. So medical application of red blood cells, anemia, This is a pathological condition characterized by blood concentrations of hemoglobin below normal values. It is also possible for the number of cells to be normal. Take note of this, to be normal, but for each cell to contain a reduced amount of hemoglobin. So if you have a count of red blood cell, it's normal, but you are anemic because it carries less high oxygen. Hypochromic anemia, that's the term. May be caused by loss of blood, hemorrhage, insufficient production of erythrocytes by the bone marrow, production of erythrocytes with insufficient hemoglobin, usually related to iron deficiency in the diet, and accelerated destruction of blood cells. So these are the reasons why you have anemia, okay? Anemia is a decreased number of erythrocytes in the blood. Erythrocytosis or polycythemia is the opposite, an increased number of erythrocytes. This may be, okay, increased physiological adaptation in people who live at high altitudes where oxygen tension is low. Poly means many, and chitos is self. Okay, so that is your polycythemia. This is often associated with diseases of varying degrees of severity, increase in blood viscosity. When severe, it can impair circulation of blood through the capillaries, better characterized as an increase in hematocrit or an increase, okay, volume occupied by erythrocytes. Erythrocytes with diameters greater than 9 microns are called macrocytes. Those with diameters less than 6, they are called microcytes. Now, another abnormality is anisocytosis. Aniso, uneven, and chitos is cell, no? Red blood cell. The presence of a high percentage of erythrocytes with great variations in size. Oh, another medical application of red blood cell, you have the sickle cell disease. There is an inherited alterations in hemoglobin molecules caused by a mutation of one nucleotide. Okay, where you have there the point of mutation in the DNA of the gene for the chain of hemoglobin. The triplet GAA or for glutamic acid is changed to GUA, which specifies valine. A result of this, the translated hemoglobin differs from the normal type of the presence of valine in the place of glutamic acid. So there is an exchange. Instead of glutamic acid, you have valine. Okay. When the altered hemoglobin, called HbS, altered hemoglobin, is deoxygenated, okay, as it occurs in venous capillaries, it polymerizes and forms rigid aggregates that give the erythrocyte a characteristic sickle shape. Okay, sickle shape, that is, you know, the sickle, that is the communist China, diba? May sickle. The sickled erythrocyte is inflexible and fragile. So, wala siyang flexibility at madali siyang masira. Has a shortened lifespan that leads to anemia. And of course, increases the blood viscosity. Okay, maano siya. Ano ang viscous? Okay, yung viscous, yung malapot. Can damage the walls of blood vessels, promoting blood coagulation. Blood flow is retarded or even stopped, leading to severe oxygen shortage, anoxia in the tissues because it cannot pass through to the tissues. It cannot give off the oxygen. Okay. There you are. Now, another medical application is hereditary spherocytosis, disease of the erythrocytes, characterized by spheroidal erythrocytes that are more vulnerable to sequestration and destruction by macrophages, causing anemia and other symptoms. This is related to deficiency or defects in the spectrin molecule. The spectrin molecule is seen in your plasma lemma, no? Okay, in the cell wall of your red blood cell. You will see it later. Surgical removal of the spleen or splenectomy usually ameliorates the symptoms of hereditary spherocytosis because the removal of a large portion of the body's macrophages. As you know, you have a lot of macrophages in your spleen, okay? And therefore, removal of these eh, will better off patient with hereditary spherocytosis. Okay, now let's go to leukocytes or white blood cells. Okay, these leukocytes migrate to tissues where they perform multiple functions. Okay, most die of apoptosis. Leukocytes are divided into two groups according to granules and shape of nuclei. So you identify them by the granules, okay, the color perhaps of the granules under certain dyes that you use, and the shape of the nuclei. You have granulocytes, okay, polymorphonuclear leukocytes. Agranulocytes, they are the mononuclear leukocytes. Both granulocytes and agranulocytes are spherical white while suspended in blood plasma. Some become amoeboid after leaving the blood vessels and invading the tissues. Granulocytes possess two types of granules. You have the specific granules. These bind neutral, basic, or acidic components of the dye mixture and have specific functions. You have the other one, which is the azurophilic granules. They are stained purple and are lysosomes. Granulocytes have nuclei with two or more lobes. Okay, this include the neutrophils. Basically, the neutrophils have more than two, up to five. And then eosinophils, you have two, okay, lobes. They are bilobes, and they have different color of granules. And the basophils. All granulocytes are non-dividing terminal cells with a lifespan only of a few days. Okay, so the resulting death is cleared by the macrophages. If there is a cell death in a particular area, then it is cleared by our macrophages that elicits an inflammatory response. We will go each one of the leukocytes. Okay, the leukocytes, granulocytes, differentiation. Being non-dividing terminal cells, granulocytes do not synthesize much protein. Okay. Golgi complex and rough endoplasmic reticulum are poorly developed. They have few mitochondria. Okay. So they have low energy metabolism. And then they depend their activities on more on glycolysis. They contain glycogen and can function in regions scarce in oxygen, such as inflamed areas. They do not have specific granules. They contain azurophilic granules. These are the lysosomes that bind the azure dyes of the stain. The nucleus is round or indented. Okay. Examples are your lymphocytes. You have your lymphocytes, and then you have the monocyte. Monocyte is big, the biggest of all your white cells. Let's talk about the first one, which is neutrophil. You know the normal value of this is 60 to 70%, or say 65 to 70%. It is 12 to 15 microns in diameter in blood smears. With a nucleus, usually 3 lobes, okay? So linked by fine threads of chromatin in females, the inactive X chromosome appears as a drumstick-like appendage on one of the lobes of the nucleus. So this would be your drumstick, okay? The one that you see here, the drumstick. And this is your neutrophil, okay? They have more than 2, up to 3, mainly up to 5, but mainly 3 lobes, okay, of the nucleus. So when you are asked, where do you find your bar body? The lobes of the nucleus, that's in neutrophil. And this is, if you identify this, basically it carries the X chromosome. X, okay. So this characteristic is not obvious in all neutrophils in a blood smear. You can see this only in some few. If you are a good, a med tech or one who reads the neutrophils or blood. The more abundant granules are the specific granules. The second granule population in neutrophil consists of azurophilic granules. Azure meaning they love. Philic granules, which are lysosomes, 0.5 in diameter. Okay. Neutrophils also contain glycogen in their cytoplasm. Glycogen is broken down into glucose to yield energy via the glycolytic pathway of glucose oxidation. So this is their source of energy. The citric acid cycle is less important. So what is more important is the glycolytic pathway. Less is the citric acid cycle, okay? As might be expected in view of the lesser number of mitochondria, because in the citric acid cycle, you make use of the energy given off by the mitochondria. The ability of neutrophils to survive in anaerobic environment is highly advantageous since they can kill bacteria and help clean up debris in poorly oxygenated regions like your inflamed or necrotic tissue. The short-lived cells, the neutrophils, with a half-life of 6 to 7 in blood and a lifespan of 1 to 4 days. They're active phagocytes of bacteria and other small particles. Neutrophils are inactive and spherical while circulating, but show an active amoeboid movement upon adhering to a solid substrate, such as collagen in the extracellular matrix. Remember, collagen, okay, the most common in the extracellular matrix. Okay, now, neutrophils may be pictured in some of your smears as in band forms, okay? They are the new neutrophils that have gone from their bone marrow to the circulation. They are horseshoe. Their nucleus is horseshoe shaped. There's an increased number of band neutrophils in the blood indicates a higher production of neutrophils, probably in response to a bacterial infection. So in an example, if it is case question, then you will be asked if there is an abnormal increase of neutrophil or white blood cells, and you see its cell with which is increased, is it eosinophil, is it neutrophil? So if it is neutrophil, then you think of a bacterial infection. If you think perhaps of an allergic reaction or a parasitic infection, then you will see more of eosinophilia. Neutrophils with more than 5 lobes are called hypersegmented, okay, and are typically old cells of the neutrophils. Oh, there you are. There is what you call the neutrophils. Look for bacteria to engulf, okay, phagocytosis. They start with pseudopodia and internalize them, okay, in vacuoles called phagosomes. So in-engulf nila by the use of their vacuoles, whose membrane is derived from the neutrophil plasmalemma. Okay. Immediately thereafter, specific granules fuse with and discharge the contents into the phagosomes where they are destroyed now. Okay, so phagocytosis. Okay, by means of proton pumps in the phagosome membrane, the pH of the vacuole is lowered to about 5. A favorable pH for maximal activity of lysosomal enzymes. As you know, we have mentioned already the lysosomes present. So when the bacteria or debris have been engulfed by your phagosome already, via your pseudopodia, okay, then it is destroyed because of this presence of lysosomal enzymes. Azurophilic granules then discharge their enzymes into the acid environment, killing and digesting the microorganism. During phagocytosis, a burst of oxygen consumption leads to the formation of superoxide anions. This is actually not 22, it's only oxygen like O2 anions. They are what you call the short-lived, highly reactive free radical that together with the mucopolysaccharides and halide ions form a powerful microbial killing system inside the neutrophils. So this is what you call your superoxide anions. And then you have, of course, your hydrogen peroxide. Okay, here, hydrogen peroxide. This is superoxide anion. This is short-lived free radical formed by the gain of one electron by oxygen. It is highly reactive radical that kills microorganisms ingested by neutrophils. Together with myeloperoxidase and halide ions, it forms a powerful killing system. Myeloperoxidase, and yeah, that's right. Dysfunctions of phagocytosis, you see. Okay. You are expected that your neutrophils have a normal phagocytosis system, but again, you find at least two dysfunctions. Actin does not polymerize normally, and the neutrophils are sluggish. So therefore, they cannot act on the bacteria, pathogenic perhaps, or the intruders, you know, the debris that are present because they are sluggish. And there is a failure to produce this oxygen, okay, this hydrogen peroxide, the oxygen that is superoxide anions, hydrogen peroxide and hypochlorite, and microbial killing powers is reduced, okay? And you see this second condition wherein the phagocytosis is defective in the chronic granulomatous disease, the granuloma. When you go to second year in pathology, then you will meet these different conditions. This dysfunction results from a deficiency of NADPH oxidase leading to a deficient respiratory burst. Of course, NAD or nicotinamide adenosine dinucleotide phosphate, NADPH, leading to a deficient respiratory burst. Children with these dysfunctions are subject to persistent bacterial infection. They are easily, you know, infected, okay, especially the upper respiratory. More severe infection result when neutrophil dysfunction and macrophage dysfunction occur simultaneously. Okay, eosinophils. We go now to the second after your neutrophil. We have eosinophils. The main identifying characteristic is the presence of many and elongated refractile specific granules. Okay, it's about 200% that are stained by eosine. So therefore, you have the arrows pointed. Okay, okay, this one here. Okay, this one, this one. Eosinophilic specific granules have a crystalline core, you call them internum, that lies parallel to the long axis of the granule. You call that, okay, internum. It contains a protein called the major basic protein with a large number of arginine residues. This protein constitutes 50% of the total granule protein, accounts for the eosinophilia of these granules. The function of this is killing of parasitic worm. Okay, so when you have eosinophilia, think of parasites. The less dense material surrounding the internum is known as the externum or the matrix. These are the less dense, okay, surrounding the internum, okay. Okay, the medical application. An increase in the number of eosinophils in blood, eosinophilia, is associated with allergic reactions and helminthic or parasitic infections. In tissues, eosinophils are found in the connective tissue underlying epithelia of the bronchi, gastrointestinal tract, uterus, and vagina, and the surrounding parasitic worms. These cells produce substances that modulate inflammation by inactivating the leukotrienes and histamine produced by other cells. They also phagocytize antigen-antibody complex. Corticosteroids, hormones from the adrenal cortex, produce a rapid decrease in the number of blood eosinophils, probably by interfering with their release from the bone marrow into the bloodstream. Far less numerous than neutrophils, constitute only about 2 to 4% of the leukocytes in normal blood, about the same size as the neutrophil, contains the characteristic bilobe nucleus. So in identifying eosinophil, first you look at the lobe. If it is bilobe, therefore, okay. Now you look at their granules, okay. Hematoxylin eosin is the pinkish granules there. So this is your eosinophils. Now let's go to basophils. Made up of less 1% of blood leukocytes and are therefore difficult to find in the smears of normal blood, less than 1%, but 12 to 15 microns in diameter. The nucleus is divided into irregular lobes, but the overlying specific granules usually obscure the division. So you have here, say, this nucleus, okay, but then because of these different granules, they are not easily identifiable, okay, the division. The specific granules, 0.5 micron in diameter, stain metachromatically, meaning to say, change the color of the stain used with the basic dye of the usual blood stains. This metachromasia is due to the presence of heparin. Okay, there you are. The basophils produce heparin, and because of heparin, they are metachromatic. Specific granules in basophils are fewer and more irregular in size and shape than the granules of the other granulocytes. Basophilic specific granules contain heparin and histamine. Basophils may supplement the functions of mast cells in immediate hypersensitivity reaction by migrating under special circumstances into connective tissues. So in hypersensitivity reactions, you see basophil, although you have less 1% of it. Basophils, there are similarity, as you have read perhaps in the previous slides, between granules, okay, of basophils and those of mast cells. Okay, so basophils and mast cells. Both are metachromatic and contain heparin and histamine. Metachromasia is a property that enables certain substances to change the color of some basic dyes, being stained by the changed color, purple, in this example, features of our basophils. Basophils can liberate their granule content in response to certain antigens, as can also mast cells. So these are their similarities. Despite the similarities, they present mast cells and basophils. They have the same, for even in the same species, they have different structures and they originate from different stem cells in the bone marrow. So there are two distinct cells, although they have similar contents. Okay, your have basophils, they have your granules and histamine, but they have different structure and they originate from different stem cells in the bone marrow. Okay, let's go to the lymphocytes. The lymphocytes constitute a family of spherical cells with similar morphological characteristics, classified into several groups according to distinctive surface molecules or markers, which can be distinguished by immunocytochemical methods. They have diverse functional roles. They are related to immune reactions in defending against invading microorganisms, foreign macromolecules, and cancer cells. There are, it is it has a spherical nucleus. Sometimes there is an indentation. This is an indentation. There you are. Chromatin is condensed and appears as coarse clumps so that the nucleus is intensely stained in the usual preparations, a characteristic that facilitates identification of lymphocytes. The nucleolus of the lymphocyte is not visible, but it can be demonstrated by spatial staining techniques and with an electron microscope. Okay, so there you are. The lymphocytes, the cytoplasm of the small lymphocytes is, scanty. It appears as a thin rim around the nucleus. Okay, slightly basophilic, assuming light blue color in stained smears, contain a few azurophilic granules. The cytoplasm of the small lymphocyte has a few mitochondria and a small Golgi complex. It contains 3 polyribosomes. Lymphocytes, you have 3 types. Small lymphocytes, lymphocytes with diameters of 6 to 8 microns, predominant in the blood. Next one is the medium-sized lymphocytes, few number, and the large lymphocytes, diameters up to 18 microns, present in the circulating blood. This difference has functional significance in that some larger lymphocytes are believed to be cells activated by specific antigens. So large lymphocytes are believed to be activated, cells activated by specific antigens. The varying lifespan. Some live only a few days, others survive in the circulating blood for many years. That's why sometimes when you are sick, you have these lymphocytes, chronic illness, you see lymphocytes. Okay. Lymphocytes are the only type of leukocytes that return from the tissues back to the blood after diapedesis. Diapedesis, huh? You should know the meaning of this. I don't like that, Mr. Tumenyo. Aldwin, please change your picture there. You are not lobo ba yan? Okay, ay, ito pa, isa pa. Okay, bear or whatever. Picture nila. Okay, next is the monocytes. Bone marrow derived granulocytes. You have with diameters of 12 to 20, it's the biggest, okay? The nucleus is oval, okay, horseshoe or kidney shape, and is generally eccentrically placed, okay? So this is example of kidney shape, or this is more of a horseshoe, okay? The chromatin is less condensed than that in lymphocytes, okay? The chromatin granules are less dense and less, okay. Because of their delicate chromatin distribution, the nuclei of monocytes is stained lighter than those of large lymphocytes. Okay, if this is light blue, then perhaps the lymphocytes would be dark blue or violet, okay? So this is your monocytes. Cytoplasm of the monocyte is basophilic and frequently contains very fine azurophilic lysosomes, granules, okay, giving it a bluish-gray color in stained smears, okay, bluish-gray color, okay, bluish, this one here, okay, gray color in stained smears. In the electron microscope, 1 or 2 nucleoli are seen in the nucleus, and a small quantity of rough endoplasmic reticulum, polyribosomes, and many small mitochondria is observed. A Golgi complex involved in the formation of the lysosomal granules is present in the cytoplasm. Many microvilli and pinocytic vesicles are found at the cell surface, microcytic microvilli. Blood monocytes are not terminal cells. They are precursor cells of the mononuclear phagocyte system. So it is not a terminal cell. As I mentioned earlier, before in our first lecture, monocytes, they are circulating, okay, but they become precursor of the mononuclear phagocyte system, okay. When they are assigned to a particular liver or to a particular organ, then they have a name, okay, a change in name, and like in the lungs also. After crossing veneal or capillary walls and entering connective tissues, monocytes differentiate into macrophages. There are monocytes who are circulating in the blood, but when they enter the organ, entering into the connective tissues, they form or differentiate into macrophages. So as I said, in the liver, they become Cooper cells, while in the lungs, they are called dust cells. Okay. Okay. Then platelets, they are non-nucleated, okay, disc-like cell fragments, 2 to 4 microns in diameter, originate from the fragmentation of giant polypoid megakaryocytes that reside in the bone marrow. Promote blood clotting and help their gaps in the walls of the blood vessels, preventing loss of blood. That's why when you have injury or you cut yourself, there are blood oozing, yeah, these platelets go there fast and they sort of seal it. So you have already the clotting, okay? And the normal platelet count range from 200 to 400,000, okay, per microliter of blood. Platelets have a lifespan of about 10 days. 10 days. Okay, now the platelet functions. Role of platelets in controlling hemorrhage can be summarized as follows. Primary aggregation, okay, and resulting to a platelet plug. And then you have the secondary aggregation, okay. Platelets in the plug release an adhesive glycoprotein, an ADP, and therefore there you will have an increased size of the platelet plug, and therefore the bleeding is stopped. Okay, of course, the third step, okay, you have your blood coagulation. Okay, we mentioned this already last time. Clot retraction, okay, of the clot. Therefore, you can see now it's clear, okay. When the clot removal, protected by the clot, the vessel wall is restored by new tissue formation. The clot is then removed mainly by the proteolytic enzyme plasmin, okay. Enzymes released from platelet granules are contribute to the clot removal. So you have your primary aggregation, secondary aggregation, you have your blood coagulation, clot retraction, and clot removal. Okay. The medical application, again, you have hemophilia A and B are clinically identical, differing only in the deficient factor. Both are due to sex-linked recessive inherited disorders. Blood from hemophiliac patients does not coagulate normally. The blood clotting is prolonged. Persons with this disease bleed severely even after mild injuries, such as a skin cut, and may bleed to death after more severe injuries. The blood plasma of patients with hemophilia A is deficient in clotting factor 8 or contains a defective factor 8, one of the plasma proteins involved in fibrin generation. 8, therefore. Okay, hemophilia A, 8. In hemophilia B, the defect is in factor 9. In severe cases, the blood is incoagulable. There are spontaneous hemorrhages in body cavities, such as major joints and the urinary tract. Generally, only males are affected by hemophilia A. That's why we have, if you're reading the hemophiliacs, the Romanovs or the British crown, they have their ancestors with hemophilia because the recessive gene to factor 8 is in X chromosome. X chromosome. So if it has X, XX is what? Female, and XY is male. So this the X carries the factor 8. Females may have one defective X chromosome, but the other one is usually normal. So that is XX. Females develop hemophilia only when they have the abnormal gene in both X chromosomes, a rare event. However, women with a defective X chromosome may transmit the disease to their male children. So they are the carrier. Women do not have this disease, hemophilia, but they carry it into their male children or descendants. Okay, let's go lymph. This is very short. Fluid medium of the lymphatic vessels, similar to plasma but with low concentration of proteins and with high amount of cells, especially lymphocytes. It is a clear liquid except for those from the small intestine which become milky. You call that chyle. After meal, okay, chylomicrons from digested lipids. So that's chyle. And generally, of course, it is clear fluid, your lymph. Okay, it drains excess fluid. Okay, drains excess fluid, lymph, from the extracellular spaces and returns it to the blood vascular system. Because if the lymph is not returned to the blood vascular system, there your total volume of your circulating blood will be diminished. Okay, it is also unidirectional, and its endothelium lack a continuous lamina. Okay, there you are. A presence and the presence of bulb. Okay, so you have here, okay, your bulb present, and therefore it is similar to the venous system. Okay, the veins, you know, that you have your bulbs. What's the purpose of this presence of blood, Mr. Okay, Buñales, what do you think is the purpose of this presence of bulb in your lymph capillaries? Buñales, what is, do you think, the function of this bulb?
Function of this bulb.
What? Come on.
It
Bakit ka,
drains excess fluid.
what? Playing what?
Oh, what? It prevents backflow.
Okay, that's good. Okay. Prevents backflow. There's to maintain a unidirection. Okay. Good. Okay, here you see, you have here your, okay, this is the tissue fluid. This is our circulation, the arterial and the venous. And you have here your lymph, okay, in the tissue fluid. It goes to the lymphatic capillaries, and then it goes to the lymph node where it is, you know, removed, or if there is any debris or whatever, because it must be sterile if it goes to the heart. Okay, then you have a bulb there. Okay, you have the lymphatic vessels. So you have the capillaries to the lymph nodes, and then it goes to your into the vein. You will see it later. Okay, up. And you have here the lymphatic capillaries from your lower part of the body. Again, the lymph node to cleanse it, and then goes to the heart, to the bulb. You will see in the next slide. Okay, there you are. Similarly, you have here. Okay, you see here. Okay, you have here from the intestine. Okay, and interstitial fluid. This one, the blue one here, from the interstitial fluid between tissues, or within this one here, and goes to the lymph vessel. Carry lymph vessel, the lymph node where it is cleansed. Okay, and then it goes to the circulation back. Now, in the area, you will see this clearly. Okay, this blue violet one here. Okay, this blue one here. Okay, okay, vessels by this area drain into the right lymphatic duct, while the upper, okay, the entire this area, okay, drain to the thoracic duct, and they go to your go back to the heart. So this one area here, okay, goes to the right lymphatic duct, and then this area here, one half down and there would go to the thoracic duct. Okay, so of course, you will see here you have your right lymphatic duct. There you are. Goes to the right brachiocephalic and then go to the down. Superior vena cava. Left, okay. Lymphatic duct again goes to the left brachiocephalic and then down to your superior vena cava and goes to your heart. Okay. Okay, there you are. There you are. And these are yung mga lymph nodes, kulani natin. They are safeguards so that the lymph that goes to the heart is sterile. Okay, there you are. This is the representation. Okay. Okay, we are done with my second lecture, but I will just have 5 minutes questions. Okay, let's call pwede, 5 minutes lang. Okay. Okay, let's see. Korea. Ariel, Nino?
Yes,
Is,
Doc.
where is, I'd like to see you. Paano ba ito? Para makita mo si, here.
Yes, Doc.
Yeah, okay. Let me just ask you a question. Okay. So when you study the blood smear and you find that there is a lot of eosinophils, what conditions would you think of that the patient is suffering from? When you see the blood smear, you have so many there is eosinophilia. What conditions would be, you would think of easily?
Bacterial
Okay.
infection.
Eosinophils. Sinabi ko na ngang easily, ano yun? Pag easily, you must have the correct answer, Mr. Correa. Easily, when you see eosinophilia, mataas ang eosinophilia, ang eosinophil numbers. So you think of what conditions?
Parasitic infection.
And?
Allergic reaction.
Oh, that's a good answer. Okay. Now, when you say anemia, what do you mean, Miss Famador? Famador? Yes, you are here. I see you in picture. Famador Queenie. Huh?
Yes, Doc.
Oh yeah. What do you mean by anemia? What interpretations? You don't have only one meaning of anemia. So give me one.
Blood concentration is below normal.
Blood? We're talking of anemia. What cell is involved in anemia? When you say blood, the entire cells are involved. But no, that's not true. It's only the what? The red blood cell. Huh? It's only the red blood cell, diba? Okay. And if it is anemia, you have greater or less?
Less, Doc. Less concentration of hemoglobin.
Hemoglobin. So you are changing again. We are just saying red blood cell. There are many, yeah? Okay, perhaps there is low hemoglobin. Perhaps there is low number of red blood cells. Perhaps their carrying capacity. Okay, so you're actually correct, but you should answer first the main question. Okay, red blood cells are the ones involved. Okay, we'll just have another question. So Miss from the bottom, Miss. Chu, Alfred, Mr. I'm sorry. Yes, there's Chu, Alfred. Let me see you. Yes. Okay. So, what do you think is the reason why we have defect in phagocytosis? Give me one answer, one that I lectured to you. Why we have a defect in phagocytosis? Why do you think sometimes we have a defect phagocytosis? Your white blood cell and your neutrophil cannot phagocytize well because of what? Diba?
Sluggish neutrophil.
Sluggish, yeah, that's right. Another one.
Um,
What more? Okay, Miss.
pH.
Yes,
Blood pH.
pH.
Blood pH.
Okay, the blood pH. Okay, now, Miss Saak, Jessa, can you hear me? Saak,
Yes,
Jessa.
Doc.
Okay, what did you learn? Okay, what did you learn from the shape of the red blood cell? What is the shape of the red blood cell? But one word only. What is the shape of the red blood cell? Jessa, Saak.
It has a biconcave,
What? By
biconcave shape, Doc.
concave.
Oh, it would assume a cup-like shape,
I don't
Doc, in a normal adult
what's that?
hemoglobin.
I don't understand your, no, I think it's not clear. The connection, I think there is a problem, no? Okay. Okay. So what is the
Hello?
normal? Yes, that's clearer now. Okay. What's your answer, Miss Saak? What's your answer?
Biconcave shape.
By
Biconcave shape, Doc. It would
dislike. Okay. Now, why is it so? Bakit? Ganon. Why? Why is it?
because it would provide, because
It would carry more oxygen. Simply direct to the point. Because it's like a balloon, diba? Kapag when you have a balloon that is already extensively, no, you know, round, it's already, you fill it so much air, then you cannot add more oxygen. Okay, that's why it is like a biconcave disc, no? Okay, so that when you add air, which is the oxygen, it can bring more oxygen to the tissues. So that is the shape. If it is like a balloon, then it will burst already, and therefore it will be non-effective. Is that clear, Miss Saak? Miss Saak, did you get me?
yes,
Okay.
Doc.
Okay.
Yes.
So okay,
Yes,
we end our no, our second lecture, and I would like Miss How
Yes, Doc.
tell me who are absent or not able to attend. You have any?
I everyone has attended po the lecture today.
What?
All are present po.
Are you sure?
Yes, Doc, but some are they got disconnected, but they can they ano tawag ito? They
Okay.
managed
All
to
right.
get back to the class again.
Okay, so my next lecture to you would be muscle tissue. Okay, so perhaps in muscle tissue, I will give a short quiz. Okay, a short quiz, 5 items only, very fast. And next meeting, that would be next meeting. So perhaps you could start early so that you 5 questions, but that would be referring to your previous lectures, the first and the second lecture. Is that clear? 5 only, so that I could give some extra credit to some of the students. Okay, so bye-bye. We will have you, if you like to join our Canvas orientation, that would start at 4 o'clock. So, and also a reminder that you will be wearing now your uniform after 1 month, isn't it? With your ID. So I think IDs are not yet here, but then at least the uniform. And also the pictures, okay? Take note that you have to submit in one 5x8 by an index card pictures with mask and without mask. This will be our records, because when you have mask, you become more beautiful or become more handsome. When without mask, then you're the best, because that's your natural. Okay, bye-bye, and thank you for listening. Until next time.
Thank you, Doc.
Thank you,
Thank
Doc.
you po,
Doc.
Okay.
Thank you, Doc.
Okay.
Doc.
Bye.
Doc.